Okay? Well, it's important that you understand how we read DNA so we know how to make RNA. Okay? Make sense so far? All right. Well, what are we reading on DNA? Because we're not reading the entire length of the DNA, because there's going to be upwards of 100,000 different genes on any given chromosome. Well, maybe not 100,000, maybe tens of thousands. Okay, it's different genes, some expressed, some not expressed. But for everything that goes on in the body, that the body is, the body becomes, there's a gene that codes for it. Okay? And all those genes exist in the chromosome. So we want to make a hormone. We don't want to unzip the whole chromosomal DNA just to find a section in the middle of it that codes for that hormone. So what we do is we come in with a DNA polymerase and we'll attach it to that particular area and we'll unwind and unzip right there, leaving everything else intact. We'll find 5' and we'll start copying. Okay, until we've copied the gene on the RNA, and then we'll release the RNA and we'll release the DNA polymerase, and it will come back together, twist, and now it's back together. Okay? So a gene is just a region that codes for a protein or whatever it is, and when we make it, it becomes messenger RNA, and that's called the mRNA. And this is what's going to actually leave the nucleus to come out and have the amino acids attached. Okay? We got it? Oh. All right. So here is I call it DNA. I'm sorry. It's an RNA polymerase. I misspoke. It comes in, unwinds, unzips the DNA strand of the chromosome, and now I'm able to come in and start to copy from 5' down to 3', and I make this ribbon of RNA. And then it leaves through the pore, and I'm ready to go out and make a protein. However, like your papers you're writing, you're not going to turn in your first draft. Why? Yeah, you want to edit it, make sure everything is perfect in it, right? Well, not everything that's transcribed is transcribed perfectly. And so we have to go in and edit that messenger RNA before we let it leave the nucleus. So it's a perfect strand of RNA, okay, so that we can make what we want to make and have it be exactly what we need. Okay? So we have what's called the first draft RNA, and that's this, coming out here, and then we're going to run into this area right here, and this is where we're going to snip out what we don't want. And those in the business call this a SNRP, which is SNRNP.
So when you are looking at the 5' and 3', that whatever that position is what can be transcribed to the RNA.
Correct.
Okay.
Yeah. All along that DNA that's coding for that gene. That can be 10,000 bases, that can be 100,000 bases. It just depends on what the protein is we're making. Okay, it can be 5,000 bases, whatever it is. Okay? All right. So to do this, this is just showing you making the showing you the RNA polymerase coming in, unwinding, unzipping, and we're just going to travel along. As we travel along, we're going to copy and make the RNA. Okay, I was actually going to take this one out because it's just repeating this. Okay. So just know what an RNA polymerase is. It's what unwinds and unzips the DNA. Now, go ahead.
Not that last one, but the one before, or with all of your things. What's the ATP for?
Well, we're going to get to that in just a second. Okay, I just kind of alluded to it. But to edit it takes energy. Okay? To snip it, to cut it takes a little bit of energy. So we'll get to that in a second. All right. So that tells the RNA to start here. What is it that tells the RNA to stop? Okay. Well, there's what we call a promoter and a terminator site that will say start copying here, stop here. Okay? So somewhere along here, your 5' is going to have what we call a promoter. Okay? And as we move on down through here, somewhere down here, we're going to have, down here, further down, we're going to have a site where it says stop. Okay? And as it does that, we'll have the promoter, we'll have the terminator, and now I'll have my DNA. Now, think of it like this. Have any of you ever made a movie? No, you're not even adventurous that way, huh? You've never made a movie in the bedroom, right? Okay. One you wouldn't display. I get it. Okay.
That's how you get hurt.
No wonder you weren't sharing when you had the herpes outbreak. Okay. If you're going to make a film, and you shoot 30 hours of film, okay? That's a good guy. You got 30 hours of film, but you've only got an hour and a half time slot. What are you going to do?
Edit.
You got to cut and edit, you got to take stuff out there. What do you have left on the floor is everything you edited out of that 30 hours down to an hour and a half, and now you have something that you're ready to take into the big screen and make millions of dollars, right? Well, your DNA, when it copies RNA, or RNA is copied off of DNA, sometimes we get 30 hours of RNA, when really all we want is an hour and a half. Okay? And so, yes, we have a promoter, yes, we have a terminator, and somewhere in between we have the movie. All right? Well, as we go along here, well, this just shows you the making of these proteins. Here I have my messenger RNA strand and my DNA, and what I'm coming out here with, well, here's my DNA, and I'm making my messenger RNA strand. Which strands are longer? These on the left or these on the right?
On the right.
The ones on the right. So which way is this going?
Right.
Left or right, certainly, okay? Because the shorter means I'm just getting started. The longer ones mean I'm getting closer to the terminator. Over here, I'm at the promoter, okay? Note that I'm not just making one strand of RNA. I'm making literally thousands of strands of RNA, okay? Do I just need one adrenaline? No, I need many adrenaline hormones, or the substance that becomes adrenaline hormone, so that I can have a sustained reaction, okay? One isn't going to cut. So, depending on, again, what that particular cell is designed to produce, I may have hundreds, if not thousands, of RNA strands being produced. And remember that I have hundreds of pores in the nucleus, and just waiting outside are literally thousands of ribosomes. Okay? So all of this is going to take place simultaneously, potentially, and I have bigger bang for my buck. It's the Ford theory. What's the Ford theory? Make an assembly line. I can make more product if I have one person doing a job, single job all day, and just move it down the assembly line. Okay? How Ford became rich. He didn't have one person making a single car. He had 10 people making lots of cars, and they just had a job to do. That's the same theory here. Okay? If we can get an assembly line going, we're going to be in great shape. Now, the editing comes along before I send it out of the nucleus, and this is called the snipping of exons and making introns. Okay? So here I have my gene, all the way down here. Okay? And I'm going to come along and I'm going to snip out a section, and eventually all I'm going to do is end up with, get rid of the intron, end up with my exon, and I actually have now what I really want, in terms of sending it out to be translated. Okay? Well, exon is the expressed region of the DNA. It's what we're actually going to make into a protein. The intron is the intervening region, and it's what's not going to be made into a protein. This will be recycled. This will be the movie. Okay? Wow. And that's the editing process. Now, I think this kind of shows you the scissors coming in there and snipping out and all that. Okay? Just essentially the same thing. But what you'll need to be able to do is to describe what an exon is versus what an intron is. Okay? And we have a different name for the intron here in just a second. So, yeah. So here we have that picture again. Okay? Well, out there you can see the intron makes what's called a layer. Okay? This is a loop. And in here I have this small protein called a SNRP. Okay? And in the presence of energy, this SNRP will snip, and this goes away. And what happens is this is bound up. So this exon gets with this exon, this intron goes away, and now my refined, ready to show, prime time, RNA can leave and go out and make what I need. Okay, so that's the RNA.
So are the lariats like glue?
Glue?
No, glue like they put.
No, the lariats are just pieces of the RNA I don't need. You know, I don't want them in my movie, so I'm editing them out. They were in the original gene, 30 hours of filming, but they're not necessary. They may be redundant. They may not be exactly copied or transcribed precisely the way I want it, so I'm going to get rid of it. And I'm just going to come down here and bring them together. Okay? All right. Now, what happens sometimes, now think about what's going on here. You have nucleotides, right? Sugar, the base, and, the phosphate and the base. The base is going to be adenine, thymine, guanine, cytosine, okay, and DNA. And they have to be laid out perfectly. And then the corresponding base pair comes up and pairs up with them, okay? Well, what's the likelihood of that happening perfectly every single time? Look around, folks. It hasn't happened. Okay. It's called a mutation. What it doesn't. Look around. All right. So here I have A, T, C, G, A, T, C, C, G, G. No idea what I built, but I just built it. Okay? Well, this is one side of the DNA. What would be over here? T, A, G, C,
T, A, G, G, C, C.
Okay? Now, does that happen perfectly every single time? No. Sometimes that gets omitted. Well, if that's omitted, what happens to this?
It's not there.
It's not there, and this slides together.